Hello, everyone, and thanks for the opportunity to present you some data today. I hope to. So, as said, I recently joined the Institute of Crystallography of CNR as a researcher. So I will basically present you some of our most recent data in my previous position at the National Institute of Chemistry in Ljubljana in Slovenia. And I will switch a bit the topic to structural biology and biophysics, especially for the neurotrophin family of proteins. And this is like an example of what can be done putting different techniques together. So let me start by sort of memory of the discoverer or the first and most renowned member of the neurotrophin family, which is the nerve growth factor, which was discovered by Rita Levi-Montalcini in the early 50s, and she was awarded a Nobel Prize for this discovery. And besides being a great scientist, she was also a great person, and having had the honor to collaborate with her in her lately scientific life, I always had I like to mention her really capacity to put together science and the beauty of science, especially. And if you think that of her drawings in her first papers showing how NGF was inducing the growth of these fibers, and these were really drawings by herself, and she was really showing how art and science can be really joined together to look how it is possible to investigate the beauty of life. So going back to the details, so neurotrophins are growth factors, and the neurotrophin family is composed by different members. NGF, as we said, then brain-derived neurotrophic factor, and neurotrophin 3 and 4, and in some species neurotrophin 5. And these growth factors interact with different type of receptors to induce different signaling. So basically, the receptors are of two different types: the TRK, tyrosine kinase receptors, A, B, and C, and each type of receptor is specific to a neurotrophin, or two in the case of TRK-B. And then the pan-neurotrophin receptor, which is P75, which is common of all neurotrophins. And actually, these receptors interact to give different cellular outcomes. And the activity of neurotrophins, I show here just for NGF for the sake of clarity, is really present in different cell types and in different biological events, and more and more, despite the fact that it's not a novel family of protein, it still has a lot to tell and to have a different pharmacological possible application. So there is also, beside these two main receptor types, there's also recently the receptors of the sortilin family were found to be interactors for neurotrophins. And so this is, I will not go into the detail, but it's just to give you an idea that how these different receptor can interact one to the other to induce different binding and subsequent signaling. So to give another complication to the picture, basically the point is that neurotrophins, which are biologically active homodimers, are expressed as pro-neurotrophins, so precursor proteins. And for many years, it was thought that these pro-neurotrophins were just folding agents, basically because neurotrophins are characterized by the cysteine knot motif. So in every protomer, there are 6 cysteines which are bound in 3 disulfide bridges interconnected in forming a knot. And but this is not the case. So pro-neurotrophins are actually interacting by themselves with the receptor to give specific signaling, which is usually even giving biological opposite functions than mature NGF. So this is quite a bit of a complication, but it is also the interesting part of it. And so in vivo, there is a balance between pro-neurotrophins and mature neurotrophins, and which can be cleaved either intracellularly, and so mature neurotrophin would be released in the extracellular space, or extracellularly and then be cleaved by other different pathways like the proteins of the plasmin cascade or metalloproteases. So going to some structure. So this is just to show you how the NGF interacts with its receptors from the molecular point of view. So there are different structures available in the PDB for neurotrophins. NGF is the most present and the most studied, and they are mainly X-ray structure. We contributed to with the solution NMR structures of NGF for both mouse and human origin. And the interesting fact is that in the case of recombinant human NGF, all the structures that are available are in the bound form. So the X-ray structure that are available are those in complex with the P75 or with the TrkA, or recently with the SORC-S2 receptors. So there are no Apo form of the protein. So they are still interesting. As said, they are involved in many different pathways and diseases, and really from cancer to wound healing and neurodegeneration, chronic pain. And there is a very high interest of the pharmacological industry in exploiting the effects of these proteins. So the actor of the show are really a lot, as I had briefly showed you. But of course, we know that biology is not easy, and there are for sure a bit more that we are not knowing yet. And so the question where we started from this project was, can small endogenous ligand play a role into this picture? And this came from some quite overlooked papers that over the years were showing that especially some oligonucleotides and lipids and glycosaminoglycans could interact with the neurotrophin, but there is no detail on the molecular description of this interaction, and so we really ask ourselves if there was any reason that we could investigate more. And so we focus both on the interaction of ATP with NGF and pro-NGF, because there was something that was driving our attention. So these reports in 2006 were saying that actually ATP was interacting to change the signaling of NGF. So we did a biophysical structural study on that. And so since I'm going to talk about protein solution NMR structure, I will just briefly explain how this is achieved practically. So basically, by solution NMR, the advantage is that you can look at protein in solution. And so it's a sort of looking into the dynamic of the protein. So it's like looking at, basically you can describe how the protein really moves in freely when it's free in solution. And of course, the limitation of NMR is the size of the protein you can investigate, although now the recent technological advances and the new experimental setups are allowing really to increase also this size limitation. But basically, there are three different big groups of protein that you can describe. So in general, it is always advisable to have double labeling for the protein, so both N15 and C13, which would allow a better dealing, to deal better with the problem. So for a relatively small protein, like less than 150 residues, the standard way is to use this type of experiment, which connects basically the amide protons with their relative carbon atoms. Usually for intermediate range protein, like between 150 and 250 residue, the protein tumbles more slowly into the magnet. So actually the spectrum is more crowded and the resolution gets worse. And so different strategies has to be used on double labeled proteins. So a full setup of many different spectra connecting different type of atoms of the residues is needed. And in the case of NGF, we actually are in this range because, as said, NGF is a homodimer, so altogether is 26 kilodalton. So one protomer is 150 residues, but of course, being a dimer, the property of the protein are those of a 26 kilodalton protein. So we are in this range. So if we would be in a bigger range, even more, of course, deuteration would be needed. And of course, in all these cases, it's advisable to use very high, higher fields, which would allow a better resolution. So as said, we collect 3D experiments, which basically, so if we start if we imagine to start from a bidimensional experiment, for example, connecting proton to nitrogen 15, and each atom would be represented by a spot, we of course evolve this experiment in a third dimension, which in this case would be C13, and we would have like spots into the space. So of course, the best way to analyze this is to cut into slices against, for example, the nitrogen dimension, and to analyze this further cutting into strips, where basically you can isolate one single residue. So this is to somehow to deconvolute the system in order to be able to analyze it. I will just briefly describe the main experiments that are used just to understand a bit the strategy. So the basic and most informative spectrum that we collect is the bidimensional proton nitrogen HSQC, or the respective proton carbon 13 HSQC. But let's say the proton nitrogen is really what it's giving you the fingerprint of the protein. So you would have the proton on the x-axis and the nitrogen on the y-axis, and basically every spot is an amide proton. So except proline, that of course you cannot see here, you would see all the residues of your protein chain, and some regions also would represent the epsilon nitrogens of protons of the lateral tryptophans, and you would see the lateral chain of the long amino acids. So this is really the way to describe a protein like a picture, and you can use this like also for quality control and comparison with published data and everything else. So we said that we need three-dimensional experiments to really assign not only the backbone amide proton, which are informative but not so much if you want to get more information. So you have to assign ideally the whole backbone and side chains of your protein, and so you would exploit other type of experiments like HNCA, for example, connects the amide proton with the alpha carbons of the own residue, so the I1 with the previous, which is the I-1. So in the experiment, what you would get is that if you imagine this is a slice to cut it into strips, in every strip you would get two peaks. The more intense one is the I carbon, the alpha carbon, and the less intense is the previous one. So you would then try to connect these strips like in a puzzle, and to see how you can connect the strips one to the other to reconstruct your chain. And of course, the chemical shift, so the position of the peaks on the carbon axis, would help you in understanding which type of residues you are having there. And of course, combining different type of 3D experiments, connecting different type of atoms of your residue chain, you can really reconstruct your assignment. So, for example, you have then experiments for a side chain like HCC8 toxi, which is again a through bond experiment. So you go from a certain proton through the whole carbons of the side chain, and you would then get the whole proton of the chain. And then we come to the most important experiments, which is actually the NOSI HSQC. This is again the N15. The same is for the C13, and this is a through space experiment. So in this case, what we analyze are all the protons connected to or better seen from an amide proton in space within a range of 5 angstrom. And this is very important because in every strip you would get all the NOEs, so these spots represent the interaction residues or atoms to your specific amide proton. So this gives you basically all the contacts that specific amide proton has in space with all the connecting residues in space. So this is the most important experiment you would use for the structural calculation, because of course you can go from the intensity of these peaks to the distance, and this would give you restraints for the structural calculation later on. So this is what we have done for NGF. So it was quite a challenge. And at the end of an MR structure, of course, since we are looking into solution, there is not only one structure because the protein is dynamic, it's moving. So at the end, what you get is a family of structures. And the overlaying of this family allows you by eye to really see where you have a secondary structure or where you don't have it. So which are the regions which are most flexible. Usually the loops are seen like this. This is just one of the representative structures. And you can see the termini are usually the most floppy ones because they're more free to move. One interesting feature, if you compare the solution NMR structure with one of the X-ray crystal structure, and this is just to remind you that our solution NMR structure is the first in the apoform because there was none available at that point. And so one interesting feature is that the N-terminus, for example, which is structured as helix when bound to the receptor, it's not in the case of the free protein in solution. So this points to some sort of induced fit type of interactions when you go to the ligand binding to receptors. So, of course, we started from the fact that we wanted to know something about ATP interaction. And so we did all the structure because, of course, this is important then to investigate how the ligand interact with the protein. But, of course, we want to investigate the interaction. So by NMR, there are different type of experiments you can carry out to have information about ligand binding. And basically, you can look up things from the protein point of view or from the ligand point of view. So since we were talking about assignment and residues, the protein-based experiments are basically titration experiments where you collect different N15 HSQCs of your protein, which has to be assigned, otherwise this would be useless. And you can follow what happens to the residues, so to your spots in the HSQC upon titration with different amount of ligands. And what you can see is to have an idea also the type of the interaction, because depending on how tight the interaction is, you would have different behavior of the different residues. So for example, if you are in the fast exchange regime, so for weak interaction, you would see a slow change from the free position to the bound position, and you can follow the peak moving or in your spectra. When you are in the slow exchange regime, so for tight binding, what happens is that basically you would see your free form residue intensity decreasing upon titration, and at the same time somewhere else in the spectrum you have the rising of a new peak. So in this case you can really have information on the type of interaction that is going on in the different positions of your protein. The ligand-based methods, so there are a lot of them, but I just here depict the STD, so the saturation transfer different, and the transfer NOSI experiments, which are the most used. And basically in this case, what you do is to somehow transfer by specific irradiation of the protein, the property of the protein to the ligand that is bound to the protein. So by difference of the spectrum of the free and bound ligand, so in this case we are looking at the ligand, you can understand which are the protons of the ligand which are involved with the protein, and with the transfer nosy you can also have some information of how these protons are then arranged into space. So with combining the protein-based experiment with the ligand-based experiment, basically you can have a whole picture of what is going on. And by helping then with other techniques and with molecular dynamic simulation, then you can put all this together and have the picture of what is happening. So this is what we have done for NGF, and so we have collected the STD in presence of different ions, and basically when you see the protons in the STD NMR experiment, you can say that these protons are involved in the binding. Otherwise, they would simply subtract in your spectra before and after irradiation. So we wanted to understand how strong is this interaction, because STD works fine in the micromolar to nanomolar to millimolar range, and to our surprise, ITC was showing a millimolar interaction. So this prompted us to think about a transient quinary interaction, so which is actually a quite interesting field. I mean, I was surprised to see how much the literature is really now digging into this point. So to really see how transient interaction are having an important role into the crowded environmental of the cell, because of course you have a lot of interactors, and this you cannot really have a tight binding, because otherwise this would block your protein to then act or move to somewhere else where it is needed. So going into some more details into this binding, so we did then the protein-based experiment for the titration, and by measuring how these chemical shifts are moving, so you see like shifts of the peaks, we identified those residues which were showing a higher degree of change, and they were actually localized in the same region of the protein. And if we look into the 3D nosy, so we collected the 3D nosy HSQC at the end of the titration, we saw that actually there were some new NOE coming out at the end of the titration with ATP, and they were really pointing to the binding to the interaction with some ATP protons. So we could identify those new interactions and use them to for some molecular dynamic simulation, which allowed to cluster somehow the more frequent positions of ATP onto NGF. And if we then overlay these positions, we can identify two sites, site 1 and site 2. And looking into the interaction of NGF with its main receptors, so TrkA and P75, it seems that these sites can interfere alternative with one or the other, and so this might be of a functional reason, and it is interesting to see that. I mean, the poses that we could see are really in agreement with our NMR data, so this was nice. And so where might this be important? So what is known about neurodegeneration, for example, or cellular stress, is that you have a massive release of extracellular ATP. So in this case, you would have a high rise of ATP into the extracellular space up to millimolar. So this is really where this kind of interaction might be a functional reason. And moreover, it is known that TrkA signaling, for example, is reduced in neurodegeneration conditions, and these two are really aspects that are pointing towards that. Of course, we are not. I mean, the biology is still to be unraveled and investigated more. So I will now briefly move to the pro-NGF. So what's the aspect of the other, the other part of the other side of the medal? So for pro-NGF, no data is available at all with ATP. So we were not even sure of how ATP could bind or if it does bind at all. So one complication with pro-NGF is that the propeptide is intrinsically a structure. So now this, the big field of the intrinsic and structural protein comes into play. So we had worked in the past, and we were the first actually to investigate into the structure of the pro-neurotrophins. And of course, we couldn't use high resolution techniques at that point because it was really difficult to work on it. And as you all know, I mean, intrinsically disordered proteins are really forcing to somehow reinvent our established approaches for investigation. But it is interesting and challenging to investigate into that. So looking into pro-NGF and ATP, so we first checked if there was at all an interaction. So by performing differential scanning fluorimetry, we saw that indeed ATP was inducing a shift into the melting point of pro-NGF, so for sure there was a sort of interaction. And to our surprise, STD and MR was looking very different than the one for NGF. So there was especially the adenine H2 proton, which was much more involved into this interaction than the others. So for sure there was a change in the way ATP interact with pro-NGF. And again, ITC was giving us a surprise because in this case, the KD was in the micromolar range. So for sure, a very different interaction. As said, being an intrinsically and structured protein and also quite big because it's 50 kilodalton. So we decided to look to use SACS, which in the past was helping us in really having some structural detail on the neurotrophin. And in fact, what happens was that in absence and in presence of ATP, there was a decrease in the radius of gyration. And if we reconstruct the models from the SACS data, indeed, upon interaction of ATP, there is a more compact conformation of the protein, which is reconstructed. We wanted to know a bit more, so we use also other techniques like to have information on that. And it was interesting that, for example, limited proteolysis were showing that in the presence of ATP, the protein was less prone to degradation. So for sure, it was less accessible. So these flexible arms that were more like close to the main mature part of the protein and were less accessible to degradation. So this was also interesting to somehow confirm this kind of conformational changes. And we further applied that by investigating HDX mass spectrometry, so proton deuterium exchange. And in this case, what you do is basically to exchange the amide proton with deuterium, and then you look into the back exchange, you quench the reaction, then you cut and you analyze the fragment by mass spectrometry. And in this case, you can really see which protons are more involved into the exchange, so more accessible or less accessible. And we did that, of course, in the absence and presence of ATP. And at the end, what you can have is that the identification of those peptides, which are more involved or more different in the two cases, free versus bound. So the red is the propeptide, the green is the mature NGF. And actually, it was interesting to see that these peptides were, again, like concentrated in the same part of the protein, and most of all the peptides in the mature part of NGF were quite nicely overlapping with the site 2 that we had identified in the mature protein. So these somehow pieces were coming together. We wanted also to see if ions were having a sort of effect. So we investigated magnesium, which is, I mean, the most the physiological one for ATP interactions. And in DSF, again, what was interesting to see is that in presence of magnesium, depending on the concentration, somehow the shift that we saw in the presence of ATP was going back. So like if magnesium was competing for ATP. And also in the case of STD, we saw a modulation depending on the concentration of magnesium. And this was also confirmed by ITC. We did ITC and limited proteolysis again. So we can say that actually magnesium and pro-NGF compete for ATP. So there is, depending on the stoichiometric ratio of the partners, you can shift this interaction towards one direction or the other. So putting all these results together with what we know from the biology of the system, we can say that in the case of a healthy brain, I mean, in healthy conditions, pro-NGF would normally extracellularly be cleaved by plasmin and to release mature NGF, which will have its own signaling pathway and then would be eventually cleaved by processed by metalloproteases and degraded. And this would be the normal way. In the case of the pathological conditions, we know that ATP is massively released, as we have seen also before, and at the same time, magnesium is massively decreased. So it is exactly the conditions where we found that actually pro-NGF is like more compact, so where ATP binding could be of functional significance. At the same time, in these conditions, plasmin levels are decreased. And having also the fact that the protein is less accessible to cleavage, this means that less NGF, mature NGF is around. And since metalloproteases are increased in the neurodegenerative conditions, and this was very much proven by biological experiments, you have an increased degradation. And actually, this might explain one of the open questions that were raised into the field about the fact that why this pro-NGF couldn't, for example, be not being cleaved by the metalloproteases as much as mature NGF. And this was something that was proven by biological experiments, and there was no clear explanation. So this might be a possible explanation for that. So by concluding, so we confirm new roles for ATP beyond its energy functions, and there is a very different binding mode when compared to NGF and pro-NGF, and the modulation of ions, it's important. And the most interesting fact is that really you can modulate the intrinsically disordered regions by interaction to adapt to different cellular contexts, and this is really might be a way how neurotrophin can adapt to different biological conditions. So probably this binding is not really by chance. So coming back to our show, there is for sure other actor in the show, and much is still to be unraveled, and we would like to go more into this, and I hope I could give you a sort of hint how you can really, I really always like to integrate different techniques which give you different point of view to have the whole picture. And let me finally thank the people involved in this study. So of course, my former group in Ljubljana, with all the people involved also for the data I didn't show in detail, like FTIR and molecular dynamics, and of course, the group in Trieste with whom we collaborated former, now we are direct colleagues, and our collaborator in Leeds for the EHDX mass spectrometry and the SACS and the grants that are supporting us. And thanks for your attention.